Question and answer on eukaryotic cell division, nuclear and cytoplasmic. Question. Unlike abiogenesis, when life first arose on this earth, currently all cells must come from pre-existing cells. So how can we reproduce, grow, or replace damaged and lost cells? Answer. How else but by the division of an existing cell? Question. In eukaryotic cells, the nuclear and cytoplasmic contents of the original cell must be copied and passed down to each new cell for the new cell to survive and function. Which phase of the cell cycle does this occur in? Answer. During interphase, which is separated into the G1 phase, where the cell grows and copies cellular contents, except DNA, the S phase, where DNA synthesis or replication to form sister chromatids occurs, and the G2 phase, where further growth and assembly of organelles occurs. Question. On the diagram, label one pair of 1. Sister chromatids, 2. Homologous chromosomes. Answer. Sister chromatids are the linear structures joined at the centromere formed by DNA replication that carry identical information for traits. On the other hand, homologous chromosomes, inherited from the two parents, are of the same size, have the centromere at the same position, and have the same genes but perhaps different alleles. Question. In interphase, the DNA that carries the hereditary information is coiled around proteins as nucleosomes and further coiled to form chromatin. At which cell cycle phase does the chromatin condense or coil up even further to form chromosomes? Answer. Chromatin condensation occurs during prophase to form chromosomes. Question. Is the cell shown haploid having a single set of chromosomes or diploid having two sets of chromosomes, each set inherited from a parent in sexual reproduction? And what is the diploid number of the organism from which this cell was obtained? Answer. The cell shown is a haploid cell, as none of the chromosomes has its homologue present. Homologous chromosomes have to be the same size and shape, that is, having the centromere at the same position. Since the cell is haploid, 4 is the haploid number N, and the diploid number 2N is 8. Question. Does cell A and or cell B have a diploid number of 8? Answer. Both cells A and B have a diploid number of 8, and even though the chromosomes in cell B have 2 strands, they are linked at the centromere and considered a single chromosome. Non-dividing cells have chromosomes or chromatin present as single strands, though their DNA is a double helix. Question. The haploid and diploid cells produced in the life cycle of humans and other sexually reproducing organisms result from different types of nuclear division. Name the cell division that produces clones of self and the cell division involved in the sexual reproduction of diploid organisms. Also, name the type of cell where each sort of nuclear division occurs. Answer. Mitosis produces cell clones, whereas meiosis is involved in sexual reproduction. Mitosis occurs in body or somatic cells, and meiosis occurs in germ cells to produce gametes. Question. Both mitosis and meiosis, though continuous processes, are said to be in a particular phase of cell division based on characteristic events occurring in the cell. Given that the cells shown in each slide are from mitosis, meiosis 1, and meiosis 2, though not necessarily in that order, and have a diploid number 2n of 6, state the type and the phase of nuclear division of each cell shown. Answer. A is a cell in prophase 1 of meiosis, as chromosomes are duplicated, condensing, and paired. B is a cell in prophase of mitosis, as chromosomes are duplicated, condensing, but randomly positioned. C is a cell in prophase 2 of meiosis, as the cell has a haploid number of chromosomes randomly positioned. Question: how about these three cells? Which of them are undergoing mitosis, meiosis 1 and meiosis 2, and which phase of cell division are each of them in? Answer: D is a cell in metaphase 1 of meiosis, where chromosomes are in pairs along a straight line through the middle of the cell or equator. E is a cell in metaphase of mitosis, as the cell has a diploid number of chromosomes aligned at the equator of the cell in a single file. F is a cell in metaphase 2 of meiosis, since the cell has a haploid number of chromosomes in a straight line at the center of the cell. Question: here are another three cells which you need to identify type and phase of division. Answer: G is a cell in anaphase of mitosis, since each chromosome is observed as a single strand and the cell has a diploid number of chromosomes moving towards each pole. H is a cell in anaphase 2 of meiosis, as the cell's chromosomes are in single strands and the haploid number of chromosomes are moving towards opposite poles. I is a cell in anaphase 1 of meiosis, since chromosomes are observed as sister chromatids linked together and the haploid number of chromosomes are moving towards each pole. Question: cytokinesis or cytoplasmic division is required to complete cell division and occurs by cell plate formation in plants or cell furrowing in animals. Which animation shows cell furrowing and which animation shows cell plate formation? Based on the animations, describe these two processes. Answer: A is cell plate formation, where small Golgi-derived vesicles join in a plane across the equator of the cell. Each vesicle contributes its membrane to forming cell membranes and matrix contents to forming the cell wall. B is cleavage furrow, where an indentation of the cell surface occurs and fibers pull the plasma membrane towards the cell center, pinching the cytoplasm into two lobes that are subsequently separated into two cells. Question: cytokinesis begins in which phase? A. Metaphase, B. Telophase, C. Prophase, D. Anaphase. Answer: cytokinesis begins at telophase, or the last phase of the nuclear division, where the chromosomes reach the pole, begin to decondense, and nuclear membranes start to reform. Summarizing eukaryotic cell division. 1. Cell division consists of nuclear and cytoplasmic division and is a part of the cell cycle, which also includes interphase. 2. During interphase, the cell grows and copies the cellular contents to provide each new cell with material needed for survival. The chromatin, which consists of DNA and protein, is not yet folded into chromosomes and can be used as a template for transcription and replication. 3. During mitosis, which takes place in somatic cells, the replicated chromosomes condense in prophase and line up in a single file during metaphase. Subsequently, the sister chromatids are pulled to opposite sides of the cell by cytoskeletal proteins called spindle fibers during anaphase, and the chromosome reaches the pole and decondenses during telophase. At telophase, the nuclear membrane also begins to reform. 4. Meiosis, which takes place in germ cells, consists of two nuclear divisions. Meiosis 1 is the first round of meiotic division and reduces the number of chromosome sets from 2 to 1. Genetic information is also mixed in crossing over, which occurs in prophase 1, and independent assortment, which occurs when chromosomes align at the equator in metaphase 1. In anaphase 1, the homologous chromosomes are pulled to opposite poles and reach the poles in telophase 1. Meiosis II, the second round of meiotic division, takes place in a way that is similar to mitosis, although the number of chromosomes have been halved. 5. Division of the nucleus is followed by cytokinesis. I hope doing the questions helped you recall and review the topic. If so, do give it the thumbs up and subscribe to see more genetic tutorial videos. Thank you.